Our final presentation for this evening, and that is Simone Melchionna from the National Research Council of Italy. I'll keep working on it. And he is a Gordon Bell finalist, so we're ending today with a bang. So come on over and listen to Simone. Thank you, Simone.
Okay, thank you very much for this nice introduction. I want to thank Nvidia for the kind invitation. So this talk is about this very recent project whose title is 20 Petaflop Simulation of Protein Suspensions in Crowding Conditions. It's done in collaboration with Massimo Bernaschi, here in the audience Mauro Bisson and Massimiliano Fatica from Nvidia. And I want to spend some words in explaining what a crowding condition is. So this has to do a lot with the cell. And the cell is a pretty complex environment. Okay. Okay, good, thank you. It's a complex environment made of several compartments, and this is a characteristic of the interiors of all cells, that the concentration of macromolecules within each compartment is pretty high. So in fact, we don't speak of highly concentrated suspensions, but highly crowded suspensions, because both macromolecules and the cytoskeleton occupy as much as 40% of the total available volume. So, in spite of the complexity of this intracellular environment, the role of crowding has been underappreciated for a long time. And this also due to the enormous computational challenge it involves to reproduce a system which is made of several thousands of proteins. So, crowding is a key factor in triggering several diseases, and the most important of which is the aggregation of amyloids. So, amyloids are fibrous proteins that tend to misfold and interact erroneously. They start sticking to each other in a way, and they lead to the neurodegenerative disorder of Alzheimer, which, of course, hits so many millions of people yearly. And there are other, of course, phenomena related to protein crowding, such as the aggregation of the crystalline in the lens. And so this is what leads to cataracts. And also sickle cell anemia is due to the fact that hemoglobins, other proteins, aggregate within the red blood cells. So altogether, crowding is a key phenomenon, or key condition, if you want, also regulating DNA transcription and replication because of the high packing of macromolecules and proteins within the cellular nucleus. So it is really essential for us to forecast the response, the collective response of a suspension of proteins in such high packing conditions. So let's go back to the cellular compartment, and this zoom here shows many macromolecules. And these are proteins, actually. These scaffolds here are proteins. And so the cytosol contains a large amount of these objects that continuously move and interact. And the mutual interaction exerted between these proteins has deep influence on the way they move around. But more importantly, it impacts the thermodynamics and the kinetics within the cell. So in a few words, all enzymatic reactions that are typically diffusion limited are impacted by the high packing condition within the compartment. So let's dissect for a while the elementary interactions within this suspension. If we take a couple of proteins suspended in the cytosol, we can categorize basically 4 types of basic interactions. The mechanical forces stemming from excluded volume, hydrogen bond, and electrostatic interactions typically take place within the protein and are responsible for holding together the different parts of the protein, so to conferring a certain shape, certain scaffolding to these objects here. But also interprotein interactions are responsible for giving to the suspension a certain packing. Other type of forces of thermodynamic, viscous, and polar origins instead control the interaction between the protein and the surrounding solvent. And fourth, the interaction between different fluid elements confer the fluid mechanics to the solution. So to the cytoplasm, if you want. But all in all, due to the erratic motion of proteins within this environment, a more complex type of interaction emerges, and this is the coupling between the motion of one protein and the surrounding solvent. So this is what we call hydrodynamic interactions. So in a few words, hydrodynamic interactions arise from the fact that as one protein moves in this environment, it disturbs the surrounding solvent. So if you want, these three seeds, these three proteins move somewhere in space and they disturb the surrounding cytoplasm, and such disturbance can travel a long way before hitting other proteins. So these two proteins at long distance, they talk. And this is very similar to what happens for the boats in the ocean, of course, but here we are in the microscopic world. So if you want, the same type of phenomenon happens at negligible Reynolds numbers, what we call in hydrodynamics. But in principle, the fact that you have so many proteins, so many proteins can also inhibit this long-range communication, which means that the streamlines arising from these proteins, say, stay caged within a local volume around the protein. This is what we call hydrodynamic screening. However, simulations show that this is not the case. If you want, communication is still long-range. These streamlines travel a long way before dying off. So hydrodynamic screening cannot be taken as a simplifying assumption. So this also gives us a lot of insight about how we should design the computational technique. But before doing so, I want to present our software, MUPHY, standing for Multiphysics Simulator. So because our system is intrinsically multi-scale and multi-level, we need a software that is capable of accommodating multiple actors within the same environment, computational environment. So MUPHY enables us to reproduce different fluids and different suspended particles or molecules, and we can cover scenarios ranging from microscopic to fully macroscopic ones. Okay? MUPHY is pretty eclectic, meaning that it can be it is a multi-platform software and can be used on cluster of CPUs, cluster of GPUs, or massively parallel architectures like blue jeans. And the versatility of the software stems from the fact that we can implement different types of fluids with different identities. They can be charged, uncharged, of different viscosities, and non-Newtonian, things like that, and different types of particles, like complex molecules, simple bodies, bodies of different shapes, and so forth. And also the code has a high degree of abstraction, which makes it quite independent on the complexity of the geometry. So if, say, you are simulating something under confinement. So let's go back to the protein crowding and sketch here how the technique works. So here we have a few proteins suspended in a fluid that now you don't see, but here we have a dual type of simulation engine, a double simulation engine. If you want, we have to handle separately the motion of particles which move somewhere in the continuum, and a fluid dynamics solver to handle the cytoplasm. However, this is not the end of the story. The reason is that we cannot resolve the fluid interactions, or if you want, the hydrodynamic interactions, from the microscopic scale of the atoms up to the global scale of the suspension. That would cost an enormous amount of computer time. So here we have to introduce a special cutoff. The cutoff is such that we resolve the hydrodynamic interaction stemming from communication between proteins, if you want, from the interaction that happens inside the same protein. So here we have, if you want, what I wrote here, subgrid interprotein hydrodynamics, because we know analytically the Green's function. So if you want the elementary interaction between two pairs of atoms within the same protein, from a coarse grain interprotein hydrodynamics that we handle through an explicit solver. So here is the mathematical foundation of the technique. First, I will concentrate on the particles. Particles move according to what we call a Brownian dynamics framework, which means that their dynamics is overdamped. So I don't give you many details here, but please trust me. So they're overdamped, and their motion happens according to the displacements of protein, the atom's positions. But here we have the mechanical forces that particles feel from the environment, so from atom-atom interactions, something that comes from the fact that a particle is embedded in an environment, and a hydrodynamic matrix, which is the Green's function of the interaction, that we call here Rotne-Prager-Yamakawa tensor, which has a pairwise form. And in its construction, we just include intra-protein terms. We exclude all of the inter-protein terms. So this tensor is basically a protein block. It's made of blocks for each protein, and it's very important because you need to do a matrix vector multiply to update the position. But more importantly, we have to compute a correlated noise. If you want the covariance matrix of this noise here stands from the hydrodynamic matrix. So if you want to compute this correlated noise, we have to do a matrix factorization in the end. Next, the fluid dynamics solver is handled through a technique which is called lattice-Boltzmann. So this guy moves on a slow manifold and obeys a hyperbolic equation. The populations here basically represent the probability of having the solvent, the cytosolic particle, somewhere in space and time and moving according to some velocity. On the right-hand side, we have something which comes from kinetic theory that tells us that particles collide. So these are collisions, if you want. On the left-hand side, we have streaming, the free motion, the ballistic motion of these particles. So what this equation is saying, once you project it on a mesh, is that these particles move over characteristics from one mesh point to the neighbor mesh point. Finally, particles and the fluid interact through a two-way coupling mechanism. So we have to compute this object here, which is a far-field hydrodynamic interactions, which has the phase of a drag force. And this drag force has some smeared version of the hydrodynamic velocity, which is a convolution in space. And more importantly, the whole thing has the phase of an explicit solver in time. So no iteration are required as in implicit schemes. So the architecture of MUPHY has, of course, the same of structure as shown before, because it has to accommodate two different types of engines. So one engine for the fluid dynamics solver and one engine for the particle solver. And these two objects move concurrently at every time step, and they are strictly bound, which meaning that we need to exchange a number of informations between the upper and the lower side of this diagram. Hydrodynamic quantities, fluid particle and particle fluid exchanges, and other type of things. And there are several criticalities, because this is a complex application, and we have to handle all these criticalities reasonably. It is important to notice that these criticalities are on both the upper and the lower side of this diagram, so we cannot really focus on a specific kernel. I can't hear, I'm sorry. Let's talk about it later, okay? So MUPHY has been applied in the past on a number of different bifluidics applications, as pertaining to DNA translocations under confinement, a flow of electrolytes in devices, in nano devices, or electrokinetics of different kinds. And a good project we worked on was studying the biomechanics of realistic cardiovascular systems by taking into account the intrinsic nature of blood, which means plasma and red blood cells, and constructing the global fluid pattern. So just to give you hints about the flexibility of these techniques. And so let's return to the cellular crowding, and I'd like to speak about the major challenges, algorithmic challenges we had to face. Well, at first, we had to decide the best coarse-grained parallelism, a choice that should guarantee, of course, a good load balance and on any number of tasks. Okay. However, we deal with multiple functional sections, and we need to have parallel efficiency, which can help on both the different sections of the method. So if you want for the hydrodynamic interaction as much as the other components. So because I'm running out of time, let me go fast. So this is, for example, a typical domain decomposition for particles and fluid according to simple cubes. So here you partition these quantities according to the box you live in, right? So there's not much thing to do. However, here you are basically distributing protons from different particles, particles from different protons in a disordered way. But you want to preserve the protein block structure in order to compute hydrodynamic interactions. So the way out is to assign particles belonging to a single protein to a given GPU and compute the short range, if you want, the near field hydrodynamics on that GPU. Okay, so we call this assignment phase a protein virtual transfer step. And then we compute the factorization of the dihydrodynamic matrix on site. So with this strategy, we ensure a good load balancing for both the, by having this dual, if you want, coarse grain parallelism, we have good load balancing for both the original management in simple cubes and the hydrodynamic interactions management. So this is how the protein virtual transfer works. Basically, we aggregate protein particles from the same protein and gather this data on a single GPU. This GPU is basically the same GPU that owns that protein at 0 time, and other choices, of course, are possible. And once we have done that, we compute on the single GPU the hydrodynamic interactions. So I'll show you in a moment how that works. So the communication scheme on the GPU, of course, needs to be constructed consistently. And it's important that here we have a lot of computation, data-intensive algorithms, and also we need to overlap as much as possible communication and kernel executions. So what we do here, we don't serialize calculation and communication, because of course we would have a lot of GPUs sitting idle while CPUs communicate, but we implement streams of executions in CUDA language, so that basically we have a way to communicate boundary data arising from each domain to the CPUs, and the CPUs communicate with other CPUs, and while this happens, GPUs can keep computing bulk quantities. And this is also, of course, useful because we use non-blocking MPI communications to have several CPUs talk together. So if you want, CPUs here work as communication coprocessors for the GPUs. And all calculations pertaining to fluid dynamics, particle interactions, and particle fluid exchange, and all the like happen on the GPU side. So these general solutions are pretty nice because we can now have a general software, and we had several crucial advantages in different applications. Here I mentioned 2 applications, 2010 Gordon Bell finalist. We ran on the Blue Gene 300,000 cores. 2011 Gordon Bell honorable mention on the Tsubame 2 architecture. We ran over up to 4,000 GPUs. But now we run on the Titan platform from Oak Ridge National Lab, which has about 18,000 GPUs and something like 300,000 cores. I skip here the specs. I want to speak a bit about our benchmark, if you want, our target system, which is made of 18,000 proteins from yeast called RAT1, which are pretty big proteins, made of about 666 amino acids. And all these 18,000 proteins interact together in this bath. It is important that we need to simulate such a big system because we want to avoid all possible artifacts arising from hydrodynamic signal that if you want, can affect the results of the simulation. So we need to have a system that big. And we have about 18 billion fluid voxels distributed equally over the GPUs and other types of numbers. But let me show you the performances. So this is the breakdown of performances according to the main segments, which are particle-based and fluid-based components. And we realized that 62% of this share is arising from molecular dynamics, but in particular by computing hydrodynamic interactions. Is that? Something's going wrong?
The show closes at 6 p.m., so we were butting up against that time.
Okay.
Hopefully you can join us tomorrow, if not for a formal presentation, at least come by so that people can ask you questions directly.
I want to show you the last figures.
Certainly.
So we conclude this slide. So besides the breakdown, this is the sustained performance obtained with such an application. 20 petaflops. It's probably a very high number, and 27.5 petaflops of peak performance. So these numbers demonstrate that we can simulate entire portions of cellular compartments by this technique, multi-scale technique. So thank you very much for your attention.
And thank you, Simone. And sorry about the delay in the start and butting up against the show of the close of show. But thank you for coming.